Let's take a look at cyanobacteria. They can do photosynthesis. So that means they produce oxygen. They also produce carbohydrates. Here's your ominous equation that you're taking water and carbon dioxide, and with the help of light energy and photosynthesis, the whole procedure, you can produce carbohydrates. That would be this one right here, carbohydrates. You produce some water maybe, and also oxygen. So these of course are very important products here of photosynthesis. And these cyanobacteria, they are sometimes referred to as blue-green algae, but they are not algae. They're truly bacteria. They're prokaryotes, gram-negatives. So anyway, some of them have these heterocysts that can also fix nitrogen, which is another neat feature of these. They are unicellular and more filamentous. And here are some examples. They do kind of look like as if they were algae, but they're not. So cyanobacteria, truly bacteria, prokaryotes. So here's a filamentous cyanobacterium, and then here we have unicellular ones over here, unicellular over here, and these filamentous cyanobacteria with these heterocysts. And here's another more exotic version. Here, these phyla of the Chlorobi and Chloroflexi. These guys do anoxygenic photosynthesis. That means they produce carbohydrates by photosynthesis, but they don't produce oxygen as a byproduct. They produce sulfur as a byproduct, but they still produce the carbohydrates. And so here we have the green sulfur bacteria, these phylum Chlorobi, and then the green non-sulfur Chloroflexi, and the purple sulfur and purple non-sulfur photosynthetic bacteria. Those are proteobacteria. We were just done, at least we thought we were done with the proteobacteria, right? So anyway, so here, purple sulfur bacteria. That's something you might see in your Vinogradsky column. That could very well be part of your ecosystem that you are digging up when you were taking some mud out. So, okay. Here's some selective characteristics of photosynthetic bacteria. The cyanobacteria are the ones that you really should know about. Cyanobacteria, they produce oxygen, so they're usually oxygenic. So that means they produce carbohydrates and oxygen. And then we have these other sulfur, green sulfur bacteria, purple non-sulfur, but that's mostly again in your Vinogradsky column you might see some of these species. Let's move on to chlamydia. These guys are intracellular parasites. You may have heard of the sexually transmitted disease. A lot of people just call it chlamydia. These bacteria, they do not have peptidoglycan as a cell wall material, and that makes them pretty fragile, and so they grow inside of other cells. So they are intracellular parasites. And there's actually chlamydia infections are very common. It's a very common sexually transmitted disease. We will talk about that in the pathogen groups. So chlamydia, and then we also have chlamydophila. They form these two different versions. They have these elementary bodies that are infective, and then they also can form the reticulate bodies. We'll see that on the next slide. But anyway, here are two chlamydias that are pathogens. We will talk about both of them, plus chlamydia as a sexually transmitted disease in our pathogen groups. Here we have chlamydia trachomatis, and that's the cause of trachoma and urethritis. And then we have chlamydophila psittaci. That's the cause of respiratory psittacosis, Parrot fever. That is. Here's sort of this life cycle. You can see how, so basically two different versions of the chlamydia bacteria. They reproduce inside of the inside of another cell, inside of a host cell, and they have these elementary bodies that are infectious. They look more like spores or something that are entering the cell here, and then they start reproducing. They turn into this reticulate body that then actively reproduces more of these reticular bodies. They're reproducing right here, and then you're shooting out these elementary bodies that then infect the next cells, the next host cell. So intracellular parasite with two different forms, the elementary bodies and the reticular bodies. Okay, so here will be chlamydia as an image. that has infected cells. So here the reticular body inside of this host cell right here, and here would be an elementary body that's trying to find a new host cell. It's infectious. Okay. And let's take a look at something more exotic. We have Gemata obscuri globus. I doubt that you'll ever hear about this again. It's pretty exotic, and I really don't have anything else to say about it. And by the way, I would not test you on something like this. So, okay, and then here some non-proteobacteria. They're still gram-negative. We have the Bacteroidetes. These guys are anaerobic, and we have Bacteroides found a lot of times in the mouse, large intestine, Cytophaga. They degrade celluloses in soil, kind of interesting, and Fusobacteria. They are a lot of times in the mouth of your pets, like dog teeth sometimes carry these Fusobacteria. They're also anaerobic, so both of these are anaerobic. Let's take a look at some Fusobacteria. Yeah, pretty cool. They look pretty cool, but not something that we have on a pathogen group. Okay, moving on to spirochetes. They look interesting because they have these axial filaments, or these endoflagella, what we call them. And here we have some important pathogens. Treponema pallidum, the cause of syphilis. Treponema pallidum, and of course we have that in a pathogen group, so we'll take a closer look at that. And then we have Borrelia burgdorferi, which is also going to be on a pathogen group. That is the cause of Lyme disease. Borrelia burgdorferi. Leptospira, that one we will not see again. Okay, so here's some spirochetes. You can see the endoflagellum, so axial filament or endoflagellum, same thing, sort of creating that curviness in the body of this bacterium. And up here would be a transmission ultramicrograph, so pretty cool. Deinococcus, there is one important one in here, Thermus aquaticus. This one here, Thermus aquaticus, lives in the hot springs in Yellowstone National Park. It's the source of Taq polymerase. Thermus aquaticus, the Taq comes from thermus and then the first two letters, aquaticus. This is a very important enzyme because it's extremely heat stable, and we can use it for the PCR reaction. So this one's very useful as a DNA polymerase that is heat resistant, and we can use it in our thermocycler for PCR reactions. Now that is the conclusion of our discussion of the gram-negatives. So I'm going to stop right here. So this was sort of the category of the other gram-negatives. I'm going to start a new recording for the gram-positives.